I just want to say apologies now if my internet cuts out at any point. Matt has a backup, we can screen share from his station, so hopefully this goes smoothly without any hiccups. Firstly, just a little intro to myself and Matt. So Matt and I met in 2016. We found out we were both really interested in carrion beetles. I'd done forensic science as my undergrad, had already done loads of studies putting out dead rabbits and trapping for carrion insects, and Matt was doing his MRes on carrion beetles at the time. But we can't take full credit for the scheme because Richard Wright, who's our kind of silent partner, he actually came up with the idea in 2009, produced a identification key, which went into a newsletter called Beetle News. And when we contacted Richard, we kind of just joined forces, revamped the key, and Matt and I, it's kind of our duty just to share our passion really, and just try to encourage more of you to share this passion with us, and yeah, recruit more recorders to go out there and look for these fascinating beetles. So over the last few years, we've been hosting workshops, usually in person before COVID, and generally just trying to raise awareness through social media. So that's probably how you found out about this today. Now, why sylphids? Well, we're pretty biased, we realise that, but they are a fascinating group and they're such a small family. I mean, globally, there's almost 200 species in the family Silphidae, and this is quite small when you consider it being a beetle family. So they're a nice small group to get to know. In the UK, we just have 21 species recorded from here. And they aren't just carrion beetles. So we've kind of deceived you a little bit with the title here, but there are a few curveballs. There are actually a few predatory species and a vegetarian species as well. So for such a small family, they're really diverse, and that's what makes me absolutely love this group. So hopefully you will too. Over to Matt.
Hi everyone. Sorry, I realised I didn't say hi before. So the Silphidae are mainly carrion feeders, but as Ashleigh said, there are some herbivorous and predatory species. They're known, so that, yeah, carrion being dead vertebrate carcasses. They're also, the Nicrophorus species are also known as burying beetles or Sexton beetles. And they are vital decomposers of organic matter. They also possess some incredible behaviours, which is what makes them, one of the reasons that makes them so interesting. So they exhibit biparental care for their young, which is very rare in the insect world. They bury the carcass. There we go. Yeah. To protect it from scavengers, and they will also actively eat or not feed young that beg too much for food. And there's an example. There you go. Yeah, and so they are really incredible beetles. The adults excavate beneath the carcass, and once the corpse or part of it is buried and prepared by the adults, so they prepare the carcass by removing the feathers or the fur, and then covering that in an excretion of preservative fluid that sort of goes over the corpse surface. And then they'll lay their eggs close to the food source, which is then utilised by larvae during development. Parent beetles will feed the early instar larvae with pre-digested material from the carcass. Larvae will then sort of beg for the food in a similar way to like a chick would with their mum. But they do it in a slightly different way. So they will sort of touch their legs on the mouth of the parent. But then successful offspring will require more food, but then too much begging can be costly because studies have shown that mothers may cull the offspring if they beg too much, just kill them off for being too annoying, basically.
So it's really this like amazing family setup. They find the carcass, they meet each other, have a lovely romantic first date on a rotting mouse or something, and then this family is created underground. And it's pretty special in the insect world. What makes them even more special is that they have this form of communication. So, lots of insects can stridulate, can make noise. But for these beetles, they kind of gone an extra step further. The way that they produce their stridulation is by having this file. If you have a look on the abdomen here, there's these two ridges. So this is called the stridulatory file. And then they rub the abdomen along, so they kind of move the abdomen in and out, if you imagine, so it rubs past their wing cases, their elytra. And if you have a little look, can you see my arrow, Matt? Is that showing up? Okay, good.
Yep.
So if you can just see some tiny hairs, and this is on the underside of the elytra here, and those little hairs rub past this file and produce the noise. Now, if anyone's disturbed these beetles in the wild before, you'll know it sounds like very squeaky. It's it's a real sort of squeaky noise. In the past, it was actually likened to like the noise that a disturbed bumblebee would make. You're in luck because we do have a short clip from my friend John. John previously worked at Edinburgh University on these burying beetles and has recorded a brilliant video with sound. So hopefully this works for you all. So it's pretty squeaky. I hope you can hear that. I'm going to play it again just so you can have another little listen. So you definitely know about it if you disturbed one. Now, I don't speak beetle, not yet, but there are 3 different types of stridulation that these burying beetles will produce. Firstly, you've got the defensive one, which I'm going to hazard a guess that this probably is. So that's to ward off any predators. Then you've got the kind of mating ritual stridulation. So the kind of courtship, if you like, noise. It's supposed to woo the mate. And then thirdly, you've got the defensive stridulation. Oh no, I've just said that one. You've got the parental stridulation. So this is where the parents in the nest kind of setup would be calling to the young to say it's dinner time. So yeah, you've got 3 forms of stridulation. And they have this fantastic form of communication, and they are so highly specialized, and it's all really down to this high competition that there is. So carrion is a really scarce resource. You've got lots of interested parties. You've got vertebrate scavengers like foxes. You're going to have competition from other invertebrates, other carrion insects like your blowflies, houseflies, flesh flies. They're all going to want a piece of this resource. So by burying it, they're eliminating some of that competition by concealing it, moving it away underground. But there'll still potentially be some things on the carcass. Perhaps the flies have already laid some eggs, perhaps they've already hatched into larvae. But the beetles are armed with a really feisty pair of mandibles, so they can pretty much devour some of the rivals that way. Unfortunately, if the carcass was too heavily infested with blowflies, it wouldn't be suitable for the beetles, so they'd have to go on and find another carrion, a piece of carrion. But then they've got the addition of competition from the microbes. So as Matt mentioned, they have to compete with the microbes on the carcass. They're trying to break down that rotting flesh. So they have these secretions that they coat the carcass in. So this actually has antimicrobial properties, and it actually slows down the decomposition as well, so that it helps preserve the carcass for longer and preserves all that kind of baby food ready for the family. Now, if you find these beetles, you'll probably also find some hitchhikers with them. Now, these little hitchhikers, we call them phoretic mites, and generally it is considered that they are beneficial to the beetle. So what will happen is, I like to call it the beetle bus, right? And mites will hitch a lift on the beetle bus, and that's because they're also interested in finding carrion. So they know that these beetles are going to head to carrion, and at that point they'll then hop off and go and predate on any fly eggs and larvae that are there. So because these mites are performing that, they are beneficial to the beetle, helping reduce that competition that we talked about. But there has been some studies that have shown that these mites will also sometimes predate on the younger instar larvae of the beetle. So it's a little bit uncertain, this relationship, and there is a lot of research into this at the moment. You'll also notice that sometimes you'll get beetles that are really heavily infested, and we also think this is probably going to, at some point, limit their fitness, reduce their fitness. You know, if the load is so heavy they're unable to fly, they maybe then can't successfully locate the carrion. So it's a bit of a fine line, and there's still some investigation to be done on this.
So here's some information you can find out about our recording scheme. So on the right here, you'll see the Coleoptera website, and this is our recording scheme page on there. There's some general information. You'll see our Twitter feed, and then also you can get the key from this page as well. So that's downloadable, it's free. We do also have an interactive key, but it's currently undergoing some edits, so we hope to release that relatively soon. You can find us on Twitter as well, at Silphidae UK, so you can see what we're getting up to. Obviously not a lot at the moment, unfortunately, but we do occasionally do Silphidae Sunday, where you can send in all your photos and things like that and get involved. You can send any IDs to us on there as well. We're actively IDing on the Beetles of Britain and Ireland Facebook page as well. So if you don't have Twitter, you can find us on there. Also on the website, you'll find this chart, which will show you all the species that are attracted to light. So if you run a light trap already, you'll use this chart, and you'll be able to see all the species that you might potentially get in your light trap.
So the basics, if you haven't tried to identify any of these beetles before, they are quite a good group for beginners because they're quite large as far as British beetles go. They're pretty big, and they are quite distinctive. So most of them can be recognised in the field. A few of them will require a hand lens to look at some of the finer features, but there's only really one or two that we would want to keep vouchers of and to have a proper close look under the microscope. So really it's a nice accessible family and good for beginners. So unfortunately, you tend to either recognise one of the two subfamilies, but first I'll just say that they have either got 10 or 11 segmented antennae. So it's 10 for the burying beetle variety, like this image here. They're generally dark in colour unless they are a burying beetle that's got flashy orange stripes. And some of them, like the burying beetles and a couple of others, have these truncated wing cases. And this may be familiar to you if you know of rove beetles, staphylinids, and that's because these are very, 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 very closely related to staphylinids. And then you've got the tarsal formation of 555. So this basically means that the tarsi here, the end points of the beetle legs, each have 5 segments, but we don't need to be counting those. Generally, once you get your eye in, you will be able to recognise whether or not it's a sylphid. But the family Silphidae is split into these two subfamilies. So you've got the Nicrophorinae, which have the clubbed antenna. That's burying beetles. Or you've got the Silphinae, which have the more sort of thread-like antenna. And if they are a club, it's a very, very gradual club that just goes gradually. You can just see the difference there. You'll really spot it very easily. So Matt's going to run you through all of our Nicrophorus. We've got 6 species found readily in the UK, and one which is most definitely extinct. So we're going to miss that one out today.
Yeah, so the first one up is Nicrophorus humator, black burying beetle, or also known as the Undertaker. It's sort of the most commonly encountered burying beetle in the UK. It's found in a wide array of habitats, whether there's just basically wherever there's some carrions that they can feed on. It's sort of active between April, September, and it's widespread and frequent in distribution. Next slide, Ash, please. There we go. So here are some species of Nicrophorinae that have the orange markings on them. The only species with the black antennae is Vespuloides. It's also got a completely hairless thorax. So you can see that highlighted there with the black antennae. Next is Vespulo, and Vespulo you can tell apart from all the other species by its curved hind tibia, and it's only got hairs at the front of the thorax, and not all of the way around. So this is, there it is. So you see on the front there, the hairs on the front of the thorax. This differs from Vestigator just slightly by that Vestigator has hind tibia, the hind tibia are straight, and the hairs go all of the way around the thorax. You're unlikely to see Vestigator as it's quite a scarce species. Hasn't been seen since 2018 in the Norfolk area. So next to the two that quite often get confused, Investigator and Interruptus. Investigator has the top orange band closer together than the bottom orange band. So you see it's got orange clubbed antennae, it's hairless pronotum, but the top band is more continuous than the bottom band. And the golden hairs of the abdomen are only on the final segment, not all over. The banding's not always the best way to identify this species, just because it can be quite variable. And that's where some people might get confused with Interruptus, the other species, which we'll go to now. So the front orange banding is interrupted, hence the name Interruptus, and the top band will be further apart than the bottom band. But as we say, it's really quite variable. So the best way to identify this beetle would be by the golden hairs on the abdominal segments. All of the segments have golden hairs, as opposed to Investigator only having the last segment with golden hairs. And here you'll see how different the banding for this specimen is compared to the one we have there. So you see it can be really quite variable. So always go by the hairs on the abdominal segments.
Thanks, Matt. That's a good rundown. So they really annoyingly have quite similar names: Vespillo, Vestigator, Investigator, Interruptus. It's a little bit mean of the taxonomist, I think. But once you get your eye and you see those differences, you can kind of come up with your own common names as well. Like I nicknamed Vespillo the John Wayne beetle because it's got that like cowboy stance with its legs. But yeah, they're good fun. And they are the ones that will be most frequently found at light traps. So if you run a moth trap, I'm sure you've encountered these, and hopefully our guides will be really useful to you. You'll be able to separate the species. Now for the Silphines, they're not all associated with carrion, and we're gonna run through these now. The main difference, as I mentioned before, is those clubbed antennae. So if you haven't got the strong clubs, it's going to be a Silphine. And our largest of this subfamily is Necrodes littoralis, and it's one that's actually regularly confused with the true burying beetles, and that's because of its large size, and also in identification keys it will talk about having the orange clubs. And if you're not familiar with the strong clubs of the other beetles, these, when the specimen was alive, it had very, very orange tips. If you just use your imagination, they're very, very orange tips to these antennae. So you can kind of see how they are similar, but if we compare them side by side, you can really see the difference there. So Necrodes has the common name, the shore Sexton beetle. And I think this is probably because in the past, entomologists did think that it was just associated with the shore. Now, in the UK, it is found inland. So don't think that you're not going to see this if you live inland. It is more coastal in Scotland and in Ireland, but if you're down in England, it is pretty widespread. It's one of our early spring carrion beetles. You'll find it from April right through to October. And it also is attracted to light, so it's another one that you might get in the traps there. You can also separate the sexes quite easily with this one. This is a male, and you can tell that from his really chunky hind tibia here, and the females have much slimmer hind tibia. And then we've got 3 predators in the Silphine subfamily. We have 2 species that are predatory on snails. So you can see the similarities here. We've got Phosfuga atrata, and this is our common snail hunter. It comes in 2 colour forms, black and red. And then we have Ablateria laevigata, and they are similar because they have these narrow, elongated heads. And you can probably guess what they use them for. They need them to access the flesh of the snail. So we'll just show you some pictures here. So yeah, it has this really fantastic narrowed head. Both the adults and the larvae are predatory on snails, and it's not really nice if you're a snail, but they have this fascinating behaviour whereby when they attack, they will bite the foot of the mollusk and then reduce some digestive enzymes, kind of leave it for a little bit, and the snail starts to liquefy a little bit within the shell, and then the beetle goes for the attack and starts really getting in there and feeding on it. Yeah, pretty gruesome for the snail, but yeah, they're fantastic predators. Actually, the easiest time to find the species is in the winter. We've actually just had a record in this week where somebody found common snail hunter under bark. So they are already preparing for overwintering. So they just spend the winter under logs, mossy areas, nicely shielded and protected from the weather. And they'll often cluster. You'll sometimes find several together. And as you can see from this photo, you'll get the red and black variations together as well. So definitely one to look out for over the winter. Now, the smooth snail-hunter, it is separated from the other one, as you can probably tell, because of its lack of distinguishing lines on the elytra here. It doesn't have the striae that the other one does. It also has a really domed shaped pronotum, if you can see that there. Very different in terms of distribution. So the common one is very, very common. It's widespread, found in many woodlands across the UK. But for Ablateria laevigata, this is only found in the south of England, and it has a real preference for sandy soils. So it's mostly going to be found on dunes, in quarries, also grazed pastures, particularly grazed by sheep and rabbits. It likes that grass really, really short. And we've also had quite a few records of it just being on open ground, on paths, on roads, where it's been easy for people to spot. So this is really one to look out for if you're in the south of England, and they're active from April through to September. And this is probably my favourite of the predatory ones. Look at it, it's absolutely gorgeous. It's a really striking beetle. You can't confuse it with anything else that we have in the UK. So this is Dendroxina quadrimaculata, the caterpillar hunter. So as you can probably guess, it feeds on caterpillars. It is mostly found on oak trees where it's going to be hunting its prey. So yes, we don't know for certain if it has any specific associations with particular Lepidoptera species, but obviously it's those that are feeding on oaks, and there's quite a few. It is also recorded from hawthorn, so it is worth, if you're going out doing any beating of oak and you see some hawthorn nearby, it's worth checking that too. It's an early spring species, so it is found from April through to June. And it's nationally scarce, so its distribution is a bit strange. Several localities in England, but in Scotland and Ireland, there's very few records. So if you are based in those places, do have a good hunt for this one next season. It's a really, really beautiful beetle. Then we have one of our most probably numerous carrion beetles for the UK. This is the red-breasted carrion beetle. It's Olceoptoma thoracicum. And this is ubiquitous, especially in woodlands. It's got a real preference for rotten fungi, dog poos and carrion. And it really likes the stinkhorn fungus. So if you're ever lucky to encounter one of these, you'll probably often find Olceoptoma on there as well in huge numbers. obviously because this stincon fungus is releasing those kind of decomposition scents, so really tasty for this beetle. It does feed on carrion, but it will also predate on other insects, so potentially any flies that were attracted to that as well. Yeah, as I said, it prefers woodlands. This is actually one that's not recorded from Ireland, but yeah, as I said, it is ubiquitous everywhere else. Then we've got 3 species of Silpha, which, if you haven't looked at them before, all look really similar. So we've got Silpha obscura, Silpha tyrolensis, and Silpha tristis. Now, the one that you're most likely to encounter is Silpha tristis. And this looks a little bit like the common snail-hunter. But if you look a bit closer, its head's not as narrow. It's not as narrow and elongate, and it doesn't have that shine. So the common snail-hunter has a really shiny exoskeleton, whereas tristis is very matte, and the only bits that are shining are these longitudinal lines and the striae. It also has a kind of blunt front to the pronotum, whereas Phosphigratata snail-hunter is very rounded at the front there. Now, the way to separate the 3 of these from each other is to really look at the kind of texture of the elytra. So we can zoom in a little bit more here, and we can see that for obscura, it's very, very matte, and they have very regular punctures. Tyrolensis is super shiny. You can tell from how we got the reflection here, trying to take an image. And the punctures are super irregular. And then tristis is more similar to obscura, but the shape is different here. If you see, obscura is quite rounded, and tristis is kind of sort of more squared off. Now, the difference is here, as I mentioned, tristis is the one that you're most likely to encounter. It's fairly widespread and common. Obscura and tyrolensis are nationally rare, so they're not ones that you're really likely to see, but worth being aware of. So if you do come across one of these, please run it through the key. Check out these features, and if you've got any questions, any doubts, just get in touch with me and Matt, and we will be able to help you out with those. Oh. For tristis, to be extra sure, you can also put it under a microscope and look for these shiny tubercles. So if you can see, every one of the punctures has a little shiny tubercle just above it. Now, there are a couple of rare species, and this one is Silpha carinata, and we're calling it the Great Ridge Wood Beetle, Carrion Beetle, and we think it's possibly gone extinct now. So Matt and I ran a survey a couple of years ago with Mark Arbuckle, where we got special permission to visit this site, the Great Ridge Wood in Wiltshire. It is the last known site of this species, last recorded in 1985 by Nash. It looks a little bit similar, doesn't it, to the common snail-hunter, with the shape and the striae. Actually, it looks a little bit similar to Silpha tristis as well. But it has the front of the pronotum indented here, as you can see. Now, you are very unlikely to come across this, but we do hope to run some surveys in the future to try and see if this beetle is still here. Because it's on a private site, it might be that it is surviving and doing well, so fingers crossed, we hope that it is. Then we have the Thanatophilus species. I really like these guys. So this one here you can see in the middle is Thanatophilus rugosus, fighting its way against the flies there to secure some carrion. So 2 most common species, rugosus. This is the wrinkled one, and we've given it the common name, the wrinkled death lover. And then Thanatophilus sinuatus is the smooth death lover. Pretty self-explanatory there. So the difference is that texture on the elytra. So it is very wrinkled. You have these tubercles in between the striae here, whereas with sinuatus, it's very, very smooth. And the other thing that's really interesting with these is that for sinuatus, it's easy to separate males and females. The females have this extended bit to the elytra here, these tips at the end. And this one can only be confused really with dispar, which is our scarce species. So dispar is a vulnerable species in the UK. It's been downgraded from a more critical status recently because it has a stable population in Scotland, but its distribution has retracted. Its strongholds at the moment are Loch Leven in Scotland and on North and South Uist. It also has a stronghold in Northern Ireland, one of the large lochs there. It's said to be associated with dead fish at the side of lochs. And we are keeping an eye on it. RSPB have been doing some great survey work over at Loch Leven, so the population is stable there. You are unlikely to encounter this one, but if you do, be aware that it comes in a red form and also a black form as well, which means that if you saw it, you could quite easily get it confused with Thanatophilus sinuatus. Just going to go back one so you can have a look. But the thing to look out for with this one, which I failed to put a picture in, I'm really sorry, it's in the key, is that sinuatus has a tiny little, what we call shoulder tooth. So it's very rounded on the shoulders of the elytra on rugosus and on dispar, but for sinuatus, it has a tiny little tooth. Now, annoyingly, you really need the beetle to be in the right position. It needs to be behaving. So if you can hold at the beetle with your fingers and very carefully use a hand lens to look at this feature, it will help you to be certain that it is sinuatus. But the other thing with dispar is that it has hairy elytra. The trouble with hairs is that if it's an older individual, they can abrade, they can wear off. So it's always good to check for that shoulder tooth. And we'll be really, really excited if anybody finds some Thanatophilus dispar. Yeah, please let us know and share your photos of that one if you think you have it. Now, one of my favourite silphines is probably this one, the golden beak carrion beetle. When you catch this one in the sun, it's absolutely beautiful. The golden hairs are just shimmery. Unfortunately, it used to have pest status in the UK because it likes to feed on beet crops, hence the name. So yeah, a bit of a pest. It's got a stronghold in East Anglia, where there are large crops for it to feed on, but it's now nationally scarce. There was no eradication of it, but it's just probably declined due to the dreaded P word, pesticides. It's a vegetarian, so it's both the adults and the larvae are feeding on plant material, and it's active between April and August. It's like a little teddy bear. It's a really, really gorgeous little carrion beetle. Definitely one to look out for. It's predominantly coastal, so stronghold in East Anglia, but it's found in Scotland. I found it on the dunes up here, and it's a great one to look out for. There is another species of Aclypea, which we believe is extinct now, not seen since the 1940s. It does look very different. Have a look at the key and you can see a picture of it in there. We haven't included it for you today. Just don't want to overload you and confuse you. Then when it comes to the larvae, the two subfamilies, as with the adults, are quite different. So the Nicrophorus, as you know already with the behaviour, they are predominantly underground. So they are going to be nice and protected. The parents are caring for them, fending off any predators. So they don't need to invest a lot of energy in producing a really tough exoskeleton. So they're quite fleshy looking. They have distinctive and very well-developed heads and legs and mandibles. But other than that, they are pretty squishy. And the 6 species all do look very, very similar. Then we've got the Silphinae, and these, because they're out and about, they're either, the parents lay their eggs under carrion, on carrion, and then the larvae fend for themselves. They need to be a bit more robust, so they have a much tougher exoskeleton, and some of them are obviously predatory, so they also need to be tough to be out and about hunting. And so they are sometimes confused with carabid larvae, sometimes confused with woodlice, and also lampyrids, also the fireflies, the glowworms. But really, it's very complex when you start trying to identify the larvae. It's something I'm working on at the moment. Basically, if you think you found one, you're more than welcome to send us photos and we'll try and do our best to identify it. But be prepared that we might not be able to, and that often you need to have a specimen to actually do the identification of those. But we're really interested to see what you find, so do take photos of any that you encounter. Over to Matt for trapping, because this is Matt's most favourite bit.
Yeah, it's the fun part. So how can you find them? Carrion feeding species mostly generalists in terms of habitat and are opportunistic on the appearance of carrion. The first and most important thing is just check any carrion you see. The smellier the better. So when you're walking along, I always see, unlike Ashleigh, unfortunately, in this photo, I'll always have some gloves with me. With me on a walk or anything, just in case I do come across some carrion. Best thing, turn them over, because often if a brood has started trying to bury it, or parents have started trying to bury it, then they'll be underneath. So that's the place to check. Also on larger carrion, you'll sometimes find the burying beetles will make a brood underneath. They won't be able to bury it because the carcass is too big, but they'll make like a brood chamber underneath the large carrion, and if you turn over a large carrion, you might often see them under there. Another place to find them is the light traps. Like we've mentioned before, several species are attracted to light. So yeah, they're a really good way of searching for these beetles. And then you can use baited traps as well. So you can bait some traps with some carrion. So there's a few ways you can do it. One way is burying the carrion. So this will generally work a bit better with larger carrion, so rabbits, squirrels, things like that. I saw someone was burying a deer head the other day. Interesting. So there's something you can bury the carcass, only shallow, only a short distance underneath the surface of the soil. And then you can put pitfall traps or anything else around them. And they can be there for a good month or two. So they're a continual source of bait. They should hopefully stop scavengers by burying them as well. And the great thing about these, but doing it this way, is sometimes when you come to excavate the body, you can hopefully find a live brood underneath. Which has happened to us once. It was very, very exciting. So yeah, always excavate, but you want to wait until the right time. So you do I always thought give it longer than you think. Seems to have worked for me in the past. So yeah, a good month or two. It will smell really, really bad though. I will warn you now.
But we are all used to wearing face masks now, so take your face mask with you when you're exhuming your bunnies.
One person has recommended putting Vicks underneath your nostrils. That helps as well. Another way is baited pitfall traps. So you can do this in several ways. You can do it simply just putting a cup in the ground with the top at ground level, put the carrion inside, and then checking that daily is ideal if you can. I'd always recommend aging your carrion when you do something like this, just because if you leave it without the netting like I've done there, that's mosquito netting to stop flies getting in, but you don't have to use that. So if you age it for maybe a week or two, depending on the size of the bait that you're using, then it will sort of go past the initial decomposition stage that flies and blowflies are so interested in. They will still be interested, but it will be a bit later on. So the Silphidae have got more chance to be attracted to it. In terms of bait, you can use anything really, any meat. So you can I use mice quite often, but you can use liver, chicken, fish. Fish is obviously really smelly. But yeah, whatever you've accidentally let go off in the fridge, you can use that. So then you'll see here, I often use cages to try and stop vertebrate scavengers from stealing the bait. But another way of doing that is to hang it, use hanging traps. So again, you hang them off anywhere, the side of your house. I live in a flat. I've got a hook on the balcony that I sometimes hang bait off to try and attract them in. But then there's also this baited vein trap, and are we watching the video, Ashleigh? So I forgot to ask.
No, I recommend that people go and check it out afterwards though. This is our friend Dan Asor has put together a really great little video on how to make your own vein trap. So if you're not familiar with a vein trap, check it out. But basically it's an interception trap. So you hang it in the tree. It's got several sections so that if the beetle hits it, it gets funneled down into your kind of bucket. And you can put your bait in there to try and entice more of these beetles in. And as Matt's mentioned, live trapping is the best. For entomologists, we tend to put some preservative in there because we maybe can't visit the site so regularly. But if you're able to check your traps daily, there's no need to be pulling any of your sample. Obviously, our main aim is to try and record these beetles and release them and monitor the populations. So to kind of give them a little bit of help in the traps, if you put some soil or a bit of sand or leaf litter in there, that will give them some cover from any predatory species in there. But yeah, have a look at Dan's video for a great visual.
And this is where we'd like you to submit your records. So we encourage people to submit their records on iRecord. It's what we use to verify. But if you don't have iRecord, you can email us. And that's silphidae@brc.co.uk. Is that right? I think. Mm-hmm. So you can send in your sightings on there, and we can upload them for you if you need to. But it's a great tool because it's free, which is great. And you can submit any records. You don't have to submit just your Silphidae records. You can submit birds, your garden birds, or anything else you want. And what's good about it, if you've got like a project going, a dissertation or something like that, you could submit several species in from one site on one day. So you can send a group in at once, and that's really handy. It was for me when I was submitting my MRes records. And then you can also view maps of where your records have come from. So you'll see where you've caught certain species. And if you're looking for them again, you'll be able to have a reference from your own records, which is really cool. And you could submit photos on iRecord as well. So it helps us as verifiers be sure that you're correct. So yeah, that's iRecord. It's a really handy tool.
Great thing about iRecord as well is that you're making your findings, the things that you happen upon, your observations, you're making them accessible to a much wider community. So you're not just helping the scheme, but you're helping anybody that wants to have a better idea of silver distributions, because that data flows through to the NBN, the National Biodiversity Network. But I'm sure many of you that have attended the amazing BioLinks workshops before have heard a lot about iRecord, and I'm sure many of you are users already, so we probably don't need to convince you too much, but yeah, please do share your sightings for us. As Matt mentioned, it really, really helps us if you include a photo. So for sylphids, the ideal record shot wants to be a dorsal shot, so from above, so that you can see the head with the antenna clearly, because basically we want to know what colour the clubs are on the antenna, or what shape the antenna are. The pronotum ideally should be nice and clear so we can see if there's any hairs there. And then also if you can get the abdominal segments in view as well, so that we can check that those hairs are there too. Now we know how these beetles behave once you get them in a pot. They're scrambling about, as you probably would try to do if you were a beetle, forced into a pot. So just try to get as many photos as you can from different angles. If you can get the legs in there, that's a bonus. But Matt and I have seen thousands and thousands of these images now, so we can usually tell. Doesn't matter if it's blurry, please upload that too. Like, we can tell that this is Nicrophorus vespillo, the bent-legged carrion beetle. You can see its nice curved hind leg here. It's also super, super fluffy, and you can make out that it's got those hairs at the front as well. So if you can, nice dorsal shot from above, but any side shots too, supplementary shots are great. Unfortunately, those mites that we mentioned earlier sometimes cover the markings. Don't worry about these. Sometimes there are features that we can still use to identify them. So I'll let you just have a little look at that one. You might be able to work out for yourselves what it is. So we can just see that there is a curved hind leg here. So that's indicative of Vespillo, same as the previous one. But if we wanted to see the hairs, they're totally obscured. So again, it would be really useful if you get like several different shots of one like this. So on that note, we thought we would leave you with a quiz. Not too serious, just a bit of fun to test, to see how much you've been listening, to see how well we've explained it as well. So we'll start with this one. So take it in, and then we're going to give you a poll and give you 3 options there. So pick the one that you think this species is. And Holly has wonderfully put all the tech together for us.
Just to point out, it is anonymous as well, so don't worry if you get it wrong.
Yeah, don't be worried about getting it wrong. Ooh, we have some people. Ooh, it's not as clear cut as I thought. Only 60% of you have voted, so please don't be shy. Put your guesses in now. You are doing well. And to say thank you to Liam Olds at this point for sharing his beautiful image with us. Liam does take some very good photos.
Okay, it's been a minute, so I think I'm gonna end up there.
Okay, congratulations, folks. 53% of you got it correct. This was Vespilloides. So the other options was humator. Humator is the all black species. So we can discount that one. And Vespillo, now was a little bit tricky. So that was the one with the curved hind legs, but Vespillo has orange clubsy antennae. So Vespilloides is one of our most common species, and it is the only one that has the black antennal clubs. So you can always discount it from the others because you know that it's this compared to the others because of those black clubs. So, oh, I can share the results with you so you can see. So well done to those of you who got it right. And for those of you that didn't, do not worry, you know for next time. On to the next one, Holly.
Okay, that should be a...
You can move the poll just to the side so you can see the beetle a bit better. So who might this be? It's one of our all black species. It's a bit on the shiny side. Don't forget to look at those antennae. Look at the head on this one as well. So that's half of you have posted your votes. Don't be shy.
Okay, a few more seconds and then I will end. There we go, 1 minute.
Oh, so folks, we didn't do a very good job explaining this one to you clearly. So it's actually Phosphuga atrata. This is the common snail hunter. Now the angle was a little bit mean for you, wasn't it? But it does have an elongated head. It's very shiny, which is the tip for this one as well. And it also, yeah, it has the very long thread-like antenna and a nice domed pronotum as well. Okay. So you did think it was Tristis, which is actually really helpful for us because initially when we wrote the keys and things, we didn't realise, because we looked at so many of them, how similar these two are. So it means that we can put together some more resources and really make sure that we are explaining the differences between Tristis and Phosphuga atrata a bit better. So this is good. I'll show you the results there. And then we have one more for you. So who might this be? Is it Thanatophilus sinuatus? Is it Thanatophilus rugosus? Or is it Necrodes littoralis? So again, look at those antennae. You want to look at the elytra for this one, the wing cases. We've been a bit mean because obviously the two Thanatophilus are quite similar. This is looking pretty good, folks. Okay.
Okay, 5 seconds, everyone. We need like a countdown timer on this.
Yeah, that would be good.
There we go, stop that.
Okay, brilliant. Well done, folks. Yeah, it was Thanatophilus sinuatus. I thought this was going to be the most difficult one. So this is really useful for us, isn't it, Matt? Because will really help us in future workshops. So congratulations. Well done, everybody who got that right. It is sinuatus. It's got those smooth sections between the ridges on the elytra. It's not got the kind of lumpy, bumpy bits that rugosus has. And of course, it wasn't Necrodes littoralis because, well, yeah, it doesn't have the round pronotum that Necrodes littoralis does, and it doesn't have orange tips to the antennae. But well done. Thank you, everybody who joined in with that little quiz there. I hope you learned something. I think we did. So that was great. Thank you. Okay, so can we get rid of that one, Holly? Have I done that successfully?
I think so. Has that gone off your screen now?
Yeah, there we go.
Okay.
So really all that's left is for us to say thank you so much for joining. So thanks to BioLinks for having us. We hope that this will encourage more recording, because really our aim is to inspire more and more people to record so that we can boost our recorder base, boost those number of records that we have, and ultimately learn more about what is happening with our UK sylphids. We already know from the 30,000 records that we've compiled over the last few years that there are changes. There's some good changes, there's some not so good changes for our sylphids, but ultimately we need the data behind us to know exactly what's going on with them. The scheme wouldn't exist if it wasn't for Richard, our fellow scheme member. So thanks to you, Richard, and also to the amazing people at the Biological Record Centre. So Martin Harvey, Jodie and Helen have just done so much for us, whether that's through technical support or just sheer enthusiasm. And yeah, it's really been brilliant. We've really enjoyed working with all these brilliant people. Thank you as well to encouragement from Darren Mann and Max Barkley, to our friends Ali and Spence. Big thanks to our respective institutions. So I work with National Museum Scotland, and thank you to them for allowing me to use some of our images today. And thanks to Edgehill University as well, who is where Matt did his MRes, and they hosted us for our first ever workshop, which was 30 participants. So
yeah, it was.
I remember thinking how that was a really, really large workshop and that we weren't going to manage. And then today we have like 130 of you. So yeah. Also want to say thank you to Ed Ivemy Cook, who helped design our logo, to all our amazing recorders who already submit their records. And thank you to you who will be joining the recruits. And thanks to all of the brilliant people who let us use their images today as well. So thank you for joining. We hope this has been interesting for you. Don't worry if you can't instantly ID every sylphid now. That's not the aim. We've kind of just put a taster out there for you, hopefully enticed you. And if you've got any queries with IDs, just come to me and Matt, and we will help you.
Brilliant. Thank you so much for that, guys. That was a great presentation. We're going to try and squeeze in a quick few questions now. So if anyone does have any questions, please send those to me in the chat. I will keep an eye on that now. I think I'll jump straight in as well. We had a few questions at the start of the presentation, all along the same lines. So Kate was asking, why do they eat the larvae that beg for food too much?
Yeah, it is a good question. So that research was done by Percy, Seth and Claire Andrews at Edinburgh University, where I was based for a little bit. So what happens is that the female has to allocate her resources. She only has so much energy, so much time, and she has a hungry brood of begging larvae. They found that the larger larvae are more successful, so they tend to obviously beg a little bit more and acquire more resources. But basically, if she has a brood that's too large, they tend to lay the right amount of eggs at the beginning. They assess the carcass and then will lay a suitable number of eggs based on the size. But if they haven't quite got it right, or there's some other variables, like they will kill off some of the brood so that it's a manageable load. And basically their ultimate goal is the female and male need to make sure that these offspring survive. So it's all about trying to raise a healthy brood that are going to make it through. So she's only got a finite amount of resources, and if some are begging too much, they just cull them off. Yeah, there's just not enough to go around. I've probably done that a very good explanation there, but
yeah, it was great. Thank you.
The paper for more.
Thank you. Liam was asking, why do the parents feed the larvae? Are the larvae unable to feed independently, or are their parents providing some kind of substance that the larvae would otherwise be unable to acquire?
That's a really good one as well. So it varies across the different Nicrophorus. Some are completely dependent on the adults for feeding through all 3 instars. Others are fed by the parents in the first 1 or 2, and then they feed independently for the 3rd. There are things we think that are passed through from the adult to the parent. So Matt was mentioning these antimicrobial properties. So there has been some research that's looked at these that's shown that some of those are passed on to the larvae as well. So yeah, there is some of that going on, and it's really all about ensuring survival and healthy offspring. So because it's such a limited resource, they really need to invest a lot of care into making sure that these offspring survive. That's why it's such a unique behaviour, but it's evolved because of this competition.
That's great, thank you. David Howden, you've got your hand up. Do you want to unmute yourself and ask your question?
Yeah, I can do. Thanks. Thanks for the presentation. It was very interesting. Feels like this is a manageable group as well, which is nice. Always difficult with beetles. I was just wondering, the phoretic mites that we seem to frequently have on them, is there anything about them you want us to capture or record if we observe these things?
Not for us. We don't track anything, we don't record them. To be honest, I can't identify them to species. So yeah, not us personally, but there are researchers out there who are really interested in these mites. So perhaps what I'll do is if you are on Twitter, I will put a tweet out and we can link up with some of those researchers that are out there, because this might be a really good opportunity to maybe kill 2 birds with 1 stone.
Great stuff, thank you. There was quite a funny comment in the chat from Mike Hill, who's a moth trapper, and he was just talking about how bad carrion beetles smell whenever they're in the trap. Sorry, I will read through now and look for some more questions. Oh, so Annie was asking, so both parents are involved in upbringing the young, but do the males and females have differing duties at all?
Yeah, Matt, do you want to take that one?
I can't remember, sorry.
Okay.
Yeah,
I don't want to hold
too
all
much
the
information in my head, I forget things.
They do tend to take slightly different roles. The male does a lot of patrolling of the carcass to kind of protect the resource from potential invaders or intruders. You will get other rival burying beetles coming in. You get brood parasitism and all sorts happening. It's a real drama. You get females killing other females, males killing other males. Yeah, it's all very dramatic. So the males do a lot of patrolling, but they both will preen and keep the carcass clean. So they get rid of any fungi that develops on it, and they both do the feeding as well. What happens in nature, though, is that the male will leave a lot sooner. So most of what we know about these burying beetles has been found through lab-based experiments. But you know, you've got a box of soil with a dead mouse and 2 beetle parents and a family. So it's kind of an unnatural setting in that the male can't really leave and go anywhere. But in nature, he probably would abandon and he would go off to find another mate and try to raise another brood. All about getting his genes out there, isn't it? So the female will do the majority of the feeding. She'll feed for longer, basically.
Great, thank you. Jan Miller, I can see you put your hand up. Do you want to unmute your microphone and ask your question? Are you there, Jan?
Hi, yes, sorry, trouble with the software. Yeah, I found a fabulous big black door beetle the other day in the garden, and I looked it up online, and some sources said it was a dung beetle and others said it was a burying beetle, and that's when I got confused. Can they be both?
No, I mean, when it comes down to the species, it couldn't be 2 things. But the thing is with dung beetles is there is some overlap. So you'll often get a dung beetle that is attracted to carrion. You also get carrion beetles that get attracted to dung. So there is an overlap in their habitat and their preferences. But for your beetle in particular, you would need to send somebody a photo, either us or the Scarab Recording Scheme, because it sounds like it's one of our groups. They obviously both have clubbed antenna. If they have orange tips, you're going to have a burying beetle. If it has a metallic blue underside, you're going to have a dung beetle, a true beetle.
Yeah. That was the other bit of the question. It had fantastic blue iridescence all over the underside.
There we go. It's definitely a dung beetle.
Oh, right. Right, that's really interesting. So that's a whole other group. They're not part of the Silphidae.
And it's a much bigger group, but definitely one to tackle next, because if you like carrion beetles, you're going to like dung beetles as well.
Okay, thank you very much. Excellent.
You're welcome.
Well, thank you. Right, I'm going to squeeze in one last question. We had a question from Nazreen asking, in which stage are the beetles attracted to the dead animals? Is there a certain amount of days? Is there a certain sort of smell that comes with them at a certain time, or what is it?
It's hard. It's hard to define because it can depend on the weather at the time, depend on the size of the carrion, moisture. Yeah, it's really, really variable. There's no necessarily set time. Initially, blowflies will be on there within an hour or half an hour of death. But then the beetles will come a few days after that, really. This is why I suggest about aging them, just to get it past that initial phase of decomposition, to age your bait, to get it past that initial stage of decomposition, and the beetles will come along after that. So it's hard to define a certain time period, but just I tend to keep them in like a plastic tub or something outside for a week. When you lift the lid, it's a bit smelly, but yeah, that's what I've recommended for trapping. But yeah, it's hard to define a specific time period.
No worries.
It's the sulfurous compounds that they get attracted to, and yeah, these are kind of released during like active decomposition. As Matt said, like the blowflies are in there straight away. They are far superior in their ability to actually locate carrion. The beetles take a little bit longer, so they do come in at a slightly later stage. But yeah, they can detect carrion from several kilometres away, so they're still talented.
Yeah,
they're pretty.
Still impressive, yeah. Brilliant. Right, well, that's the end. Sorry we have overrun slightly, everybody, but I hope you found that useful. Big thank you to Matt and Ashleigh as well. That was a great presentation. Really interesting as well, so I hope everyone found that useful and is going to start recording now as well. I'll send round the details. We posted the link in the chat to the recording scheme and the Twitter page, and I can send round that link as well to the video you were talking about earlier, so I'll send all that round in a follow-up email. Yeah, so that's it from us today. Thank you all for attending, and hopefully we'll see you all again soon.